welcome back to The Dish. My name is Johannes Fruehauf. I'm founder and CEO at LabCentral. And it's my great pleasure today to introduce to you Isaac Stoner, founder and CEO of Octagon. Isaac, thanks for coming and doing this with us. Let's get started by having you introduce to the audience what Octagon does.
Great. So I'm Isaac Stoner, co-founder and CEO of Octagon Therapeutics. Octagon is focused on developing new medicines for autoimmune disease. And we've made some really important discoveries behind what actually causes autoimmunity. So if we look at the immune cell populations that cause inflammation and autoimmunity, at the genetic level, these are healthy, normal, indistinguishable from normal, healthy, protective immune cells. It's external factors that lead these cells to become pathologically activated. What we at Octagon have been focused on is glycobiology or glyco-immunology. Part of the way that your immune cells do not react to your own healthy tissue is based on regulatory receptors called Siglec. So these Siglec receptors identify glycan structures, little sugar structures, that decorate all of your cells and proteins. What we've shown is in patients with autoimmune disease, those regulatory Siglec receptors are disrupted in very specific ways. And with our therapeutic agents, we can restore that normal regulatory mode and thus restore immune homeostasis in patients across a range of diseases without suppressing or compromising normal, healthy immune functioning. It's a really important new strategy in the treatment of these diseases.
For our audience to get to know you as a person, who are you? How did you end up finding yourself as a CEO of a startup company?
Yeah, so I was always interested in science, always interested in biology and math. Classic nerdy kid in high school who was doubling up on science and neglecting the arts. And ended up finding my way into engineering, specifically biomedical engineering. I naively thought that biology could be reduced to an engineering discipline. The more I learned about biology, the more I learned that biology is a messy system of kludges and accidental interactions, but have naively continued throughout my career to apply some level of sense to the mess that is biology. So I got out of undergrad with a biomedical engineering degree and had no idea what I was doing, had no idea what my kind of career path was going to be. I ended up teaming up with a gentleman who was starting a company. He was the sole founder, and he was looking for pretty much a lab tech, a research associate, to join and set up the lab and do some of the early grounding work to support some of our IP filings. So that was back in 2007. We were a team of two, which was very exciting, with a couple $1 million in seed funding. I learned a couple things. One, exactly how fast two people can blow through a couple $1 million in life sciences. And two, how fast the party can stop when in 2008 we had a major recession, and unfortunately that was the end of the company. It was the last person out the door, hit the lights. But I'd caught the bug already. I thought, this is cool, this startup thing, the genome, once we have the $1,000 genome, we'll understand all of biology. We'll finally got it to that place where it is an engineering discipline. Of course, now we have the $1,000 genome and we still don't understand most of biology. So right. Again, continued to be naive there. But that's how I got into the startup and biotech type world.
Cool. After that failure to start, or maybe
First
that
learning.
interrupted start,
yes.
And the financial crisis was a painful cut in our reality. When did you start Octagon, and what happened in between?
Right, so in between, there were a series of other startups that did a little better. We actually ended up shopping around the IP from that first company and meeting an entrepreneur called Jonathan Rothberg, who was starting a company down in Connecticut, again focused on getting to that $1,000 genome. And I joined there as employee number 10. We ended up growing to maybe 100 people. We were about to commercialize a new piece of technology, and we got acquired, so that was exciting. Left that to join two guys out of MIT who were starting a company focused on liquid biopsy for cancer diagnostics before that was cool, before it was even called liquid biopsy. And we again, through messy pivots, were able to get that to a successful exit as well. And so I was like, I've got it. I've got this figured out. Now we've had a couple of wins. You know, I'm going to start the next one, and this is going to be easy.
Was it?
No. So I ended up meeting my two current co-founders through a class at Harvard called Commercializing Science, taught by Vicky Sato, who's a luminary in the field. They had a really interesting approach towards exploring bacterial metabolism. And they thought, okay, this is going to give rise to a new class of antibiotics. We built out the whole business case for what was going to be Octagon, developing a new class of antibiotics, and ended up deciding, okay, it's not time to start the company yet. But if we are able to identify a molecule, get some IP on that molecule, advance it to the point where we have animal proof of concept, then we're going to take the plunge together. And so we did that. We ended up starting the company
What year are we talking about here?
So we met in 2015. We ended up starting Octagon right about the end of 2017 and spent about a year chasing down that antimicrobial play. Learning over the course of that is not a fundable business model. There's a huge unmet need. There's a huge opportunity for innovation to make a big difference. The only way to build a business there is on grants.
Yep.
So
unfortunately,
we figured that out, and we drew a line in the sand. We were a finalist for a $15 million CARB-X grant, which is this organization that funds this type of innovation. And so 5 finalists, 3 of those companies were going to get this large grant award. We decided, okay, you know, if we get this award, we're an antibiotics company. If not, we're doing a hard pivot into greener pastures.
Yep.
Had already started a skunkworks project in our back labs, and you know, we ended up being one of those 2 companies that didn't get that CARB-X
Yep.
award, and immediately we were an immunology company the next day. And I think that was a very smart move that we made.
Yeah, we could spend a whole episode talking about what's screwed up in the reimbursement for antibiotics world.
Exactly.
And why that model just doesn't function.
Yep.
But that's not the topic of today.
No.
You now are an immunology company. You
Yes.
now are going after autoimmune indications and re-educating the immune system. Can you explain what's the technology behind that?
Right. So this started, like all good discoveries, with an accident. We observed that B cells, first, that's the first cell population we looked at, have a very specific phenotype in patients with an active autoimmune disease flare. They have enzymatic expression levels that are novel and out of whack with what you see during normal immune activation. And we pulled that thread, and that thread led us to 2 things. One, this Siglec receptor, Siglec-2, on B cells that controls, specifically suppresses autoantigen reactivity, and the ligand, a novel ligand that's in the plasma of patients with autoimmune disease flare that interacts with that Siglec receptor in such a way that it's no longer doing its normal suppressive function. So it took a lot of work to validate that pathway. That's work that usually should be done within probably the academic setting.
Yeah.
We did that within a company.
Yeah.
Which really raises the bar for us as well. The amount of data we've had to generate to support this novel mechanism has been quite substantial. But we now have developed an antibody that blocks that interaction, shown that, again, it restores the activity of that regulatory receptor. And this is disease modifying in animals. We've now run 2 different animal models where we've shown, without damaging, suppressing, depleting, ablating any immune cells, we're able to have this really substantial disease modifying effect, which is very exciting.
Yeah, you have something developed that you call the EDGE assay.
Yes.
Can you explain what that is?
Yes, so everything we do at Octagon, again, starts with clinical samples. We've built a big clinical sample collection with plasma samples across a range of different indications, including B-cell associated autoimmune diseases like pemphigus, myasthenia, pemphigoid, T-cell associated diseases, infection. And what we have done is developed a way of looking at the glycobiology profile, specifically the glycan profile, on those clinical samples. So looking at specific proteins and what is dysregulated with these carbohydrate moieties in these clinical samples.
Yeah.
We can then use lectins to specifically isolate those proteins from patient plasma and then mass spec to identify them.
Are they appearing in classes? Because you don't want to develop an N of 1 therapy.
Well, exactly right. So again, this is where our large clinical sample collection becomes important, right? So what we have shown is, these glycan profiles on known abundant plasma proteins are perturbed in very specific ways.
Okay.
And when they no longer resemble a human glycan, but they're somewhere in between maybe a bacteria and a human, this is where the immune system, these regulatory layers, are disrupted. So then there's a whole kind of assay development piece around getting from that specific misglycosylated ligands to the regulatory receptor involved. Every immune cell population has one of these SIGLEC receptors, sometimes multiple SIGLEC receptors, that interact with glycoproteins.
You're going after one of those?
So right now our B cell program is focused on SIGLEC2, which is a B cell constrained SIGLEC. We have a second program that's focused on CD8 T cells, again a SIGLEC receptor that controls inflammatory cytokine production by CD8 T cells. And we've shown that's disrupted in a very similar way to SIGLEC2. I'm not going to tell you that SIGLEC identity yet because we know it, but that's our second program we have in development. It's focused on CD8 T cells.
Cool. And so you're going after these receptors with your therapeutic antibodies.
Exactly right.
How do you go about that? How do you start that program?
So first we needed to map the interaction of these ligands to where they're binding on these SIGLEC receptors. And then we use phage display in order to pan for binders, so novel antibody fragments that interact with the specific domain of these SIGLEC receptors. We then engineer those into full antibodies, and we're trying specifically to avoid having an ADCC effect, a cell-killing effect, an apoptotic effect on these immune cells. We're trying to develop really just mild blocking antibodies that are restoring the activity of these receptors.
And you have the talent for that in-house, or are you leveraging external vendors?
Right, so the only thing we keep in-house is the discovery side, these core assays we have. All of the antibody discovery work is done externally. We've leveraged Charles River, a number of Chinese CROs. All of the antibody engineering is done externally. All of the animal work is done externally. So we're keeping our core team very small, very discovery focused.
How big is your team now at Octagon?
We're 8 people, which is very fitting, and approaching clinical candidate stage. So we have a monoclonal that's looking like a development candidate that's ready to go into IND enabling work. That's again going to be completely done externally. We're going to bring 1 or 2 people in to support that work, but really trying to keep our core team lean, focused, and driving the innovative side. If it's a commodity assay or an animal model system or something like that I can pick off a menu at a CRO, we're going to outsource it.
Yep. Will you plan on, as you said, you're handing this DC over to an external help for further development?
Yes.
And then are you planning on going after new targets internally?
Right. So this is where our SIGLEC2 or CD22 program, this is the closest to going into IND enabling work. We're going to be taking on an antibody discovery effort for our CD8 T cell program before the end of this year, and then continuing to crank the handle on the discovery side. One thing we have sought to do is, early stage discovery. At the very earliest stage, this is kind of the highest risk work you can do. Yeah. And we've attempted to leverage partnerships to do some of that very early exploratory work. So we have signed a multi-year partnership with Novo Nordisk that's focused on some indications that they're very interested in and some different immune cell populations. But right now we have all the discovery work that we can handle internally with those 3 different efforts.
So other than Novo, are you looking to add other collaborators?
Always, yeah. And part of why we're sitting here today is we're making noise about that externally.
Right, so tell the audience here why they should be partnering with Octagon.
We have a really novel set of capabilities. We have a really important and characterized set of clinical samples, and we believe there's a lot of additional discovery to be made here. We have some hypotheses around things we could do in EOE and asthma and looking at neutrophil SIGLEC biology, and there's a huge amount of undiscovered biology here.
Good job selling, that's great. Let's hear about what's next for Octagon. What are some of your upcoming milestones or celebrations when you achieve them, and what's the timelines?
Candidate nomination is the big one.
Yep.
And so everything we're doing right now is focused on that lead B cell program and checking the boxes. So for a monoclonal antibody, there's a set of maybe 30 criteria that our antibody needs to get through, and we've probably done 20 of those at this point. So we're getting there, but sometime early next year we'll say, this is it. This is our candidate. This is what we're going to put into that expensive IND enabling work, and this is what we're going to put into patients as soon as we're able.
Do you have the financial runway to do all of that?
We have the runway to finalize our candidate for sure, and we've had early conversations with all of the kind of blue chip investors around town. Autoimmune disease has become a very much hot therapeutic area, and the milestone that everyone's looking for is that candidate nomination. You show us that you have a drug candidate that's ready for prime time. The IND enabling work is regarded as really low risk, right? It's expensive, but it's regarded as really low risk. And so the investors want to fund all the way into that early clinical development where we get a signal in a patient population.
Are you open to having individual programs with individual pharma partners potentially?
Definitely, and we're having some of those conversations now. One of the key decisions a company like ours needs to make is, can we bear to part with our most advanced program? Right? Can we bear to partner off our farthest along asset? Conventional wisdom would tell you no, unless it's a really, really big deal. And so, we're continuing to debate that.
Depends
internally.
on price, I would
Depends
say.
on price. There's a right price for everything, right?
Yeah.
But there's got to be a B at the end of the price. That,
Very good. So we've talked a little bit about the science. Let's talk a little bit about what it takes to become an entrepreneur and what are some of the learnings along the way. You've built a career now in biotech. You're also a convener of communities, right? So you're becoming a senior player in the audience that other people look up to. What would you like to have heard as your younger self? What would you give your younger self in terms of advice?
Yes. So I think about this stuff a lot, and my career path has been anything but deliberate. It's been kind of accidental stumblings. I do think working for experienced, talented company builders is very important, right? Really, there's, I think, a problem in the current entrepreneurial landscape where smart PhDs, smart postdocs immediately want to take that plunge. They immediately want to start a company without knowing really what goes into that. They have an exciting piece of science and they're like, okay, let's build something.
Yeah.
I think we should be working towards almost an apprenticeship model where you want to start a company, you should go spend a few years working with someone who knows how to start and build a company, right? I did that, again, completely by accident. The other thing I would say is, do not, when you start a company, put yourself in the CEO role. If you don't have an experienced board who's assigning that CEO responsibility, I'd say 9 times out of 10, 99 times out of 100, you're making a mistake.
Yeah.
When we started this company, I did not put myself in the CEO role either. I was in the COO role. We decided that what we would do is we'd be able to attract a veteran, experienced, multi-time winner CEO into that seat.
Yeah.
And as the years ticked by, we were unable to do so.
That didn't
so eventually I
materialize.
just, again, took the seat.
So you were your own apprentice, is what you're saying?
Yeah, exactly. Well, I'd spent some time working with some good CEOs previously.
I think that's super valuable advice, and I think something, as you said, that's not that often in this frothy climate, right, is forgotten, and then people fail, and it's disappointing.
There's nothing wrong with failing either.
Yeah.
Failing should be for the right reasons, though. It shouldn't be due to poor decision making. It shouldn't be due to lack of experience. It should be you failed because you had an idea, you pulled the thread the whole way through, and it didn't pan out.
Yeah.
Right? Absolutely nothing wrong with that.
But then you learn and you try again.
Yeah, you fail because the science doesn't pan out, though. Don't fail because of making boneheaded mistakes along the way.
I like that. What are your best hopes for Octagon, and what are your best hopes for our field of biotech?
So I want to see our drug in patients.
Yeah.
I really do. I think what we've developed is an important intervention. It's going to be a much kinder, gentler, precise maintenance therapy for these patients with autoimmune disease. It won't suppress normal, healthy, protective immunity. I think it's going to be a really a big win. So I'm optimistic that we're going to see our drug in patients, and I'm optimistic it's going to do what we think it's going to do. For the industry, I would really love to see the IPO window open up a little bit more. Some of the venture dollars that are chasing late stage things in these mega rounds start to look earlier towards innovation. I think there's a lot of good innovation that's not being funded today because of the kind of biotech winter that we have lived through for the past 18 or 24 months. I'm optimistic that next 12 months, this is where it turns around. We're going to see biotech spring. Winter's over.
I'll record you and quote you. Hopefully you're right.
I say that every 6 months. The next 6 months is what it happens.
Same. Now, I know and I admire you for being a convener of our industry also and your peers, and you've created a whole network and a series of events. Can you tell us about that and what's coming up and how's it developed?
Sure. So, like most of the things I've done in my life, it's been pretty accidental. So, probably about 8 years ago now, I started getting together some friends, maybe 8 or 10 people who are also active in the biotech industry, and we get together once a month and talk about, here's what's happening in the industry, what are you working on, what are you seeing, what do you think's going on? And people started asking to join, and so we went from 8, 10 to now almost 800 people on an email list where I'll send out my own thoughts on the state of affairs in biotech, what I think is interesting. People have started sending me job postings that I'll share in events. And we'll get together once a month, and usually about 50, 60 people will show up every month. We've started having corporate sponsors reach out and pick up the tab. And again, this is all completely minimal effort on my side. I do almost nothing. The group has really just gained its own momentum, and they seem to be really popular events. So, one of the things I've thought is missing in this ecosystem is these very informal events, really driving those informal collisions that make our industry what it is. So there are no RSVPs, there are no name tags, there's no expectations, there's no speaker lineup, there are no posters. Just show up, be genuine, make some connections, and I found that kind of format's been very, very popular.
When is the next one, and what's beyond that?
We're actually having one on October 16th. And one thing that we've started to try and do with the group is raise money for good causes. Again, there's no fees, no sign up necessary, none of that. But we are starting to see if we can do some good with this community that we've put together. So come on down October 16th. The last event was actually sponsored by LabCentral, so thank you very much, LabCentral, for picking up the tab on the September iteration.
We asked you to bring an orange object.
Yes.
And I see you brought a marine orange object.
I did. I brought this is an anchor buoy from my boat. My boat is nothing fancy. You can see it's not a big anchor buoy. I have an 18-foot Boston whaler that I love, and part of what I do with this anchor buoy is go fishing. I have a lot of hobbies. I'd be excellent at being retired. But if you're going to be fishing, most of fishing is not catching fish, right? If I'm not catching fish, I want to be not catching the biggest fish. So typically I take my tiny little boat and I go about 14 miles offshore to see if I can catch bluefin tuna. So I've been out a couple times.
I've had some of your bluefin tuna.
You have?
And it was excellent.
Yes, so a couple of years ago I was sitting on a cooler outside of LabCentral handing out tuna steaks. We had 70 pounds of tuna we had to give away. And I think there are a lot of parallels between taking a tiny boat and going bluefin tuna fishing and starting a startup. I've actually written a blog about this, but usually I'm set up out there 14 miles offshore, out of sight of land, and there are huge 70-foot sport fishing boats around me doing the same sort of thing. And I think I hook as many fish as they do. So,
that's cool.
Yeah.
Well, thank you for sharing.
Yeah.
Thanks, Isaac. It's been great.